In the early days of molecular biology, computing was an afterthought. We were tracking a few dozen protein sequences, and a standard mainframe terminal was more than enough to handle the workload. Look at a modern sequencing facility today, and the reality is entirely different. We have automated machines reading genetic code 24 hours a day, constantly generating massive petabytes of raw digital information. The databases we rely on, like GenBank and EMBL, now house hundreds of billions of nucleotides and tens of millions of distinct protein sequences. If you try to process this volume by writing flat procedural code, just a long list of top-to-bottom commands, your data pipeline will eventually buckle. The relationships between these biological molecules are too intricate for simple scripts. To accurately model biology in software, the code itself needs to structurally mimic the biology. This is exactly where object-oriented programming, specifically in languages like C++, comes in. To keep these massive biological ecosystems from collapsing under their own weight, we must rely on object-oriented frameworks to manage the complexity. The ultimate structural map for software design already exists in nature. It's the central dogma of molecular biology, the flow of information from DNA to RNA to a fully folded 3D protein. In C++, a class acts as a static blueprint. On its own, a DNA sequence is just a set of instructions, doesn't perform any actions in the cell. An object is the physical manifestation of that blueprint. The object is the specific physical protein built from the DNA instructions, ready to do work. But raw biological data is very fragile. When you have a massive bioinformatics pipeline manipulating millions of strings of nucleotides, an outside function can easily overwrite or corrupt a sequence by accident. The programmatic solution to this vulnerability is encapsulation. It builds a strict protective boundary. Think of it like a protein domain folding inward to protect its core, or a cell membrane shielding its internal machinery from the chaotic outside. Technically, C++ executes this by labeling the raw sequence as private data, locking it away. If another part of the program wants to read or edit that sequence, it has to pass through public methods, strict checkpoints that verify the request. Encapsulation serves as the software equivalent of a cell membrane. It guarantees that all of our genomic data remains pristine as it travels across vast computational ecosystems. When nature needs to solve a new problem, it rarely starts from scratch. Evolution relies on taking an existing design, duplicating it, and adding slight variations. In C++, this mechanism of evolutionary reuse is called inheritance. Look at a phylogenetic tree, where a universal common ancestor branches out into highly specialized species. So we apply that exact same taxonomy in code. We can build a base class called biosequence that holds the core rules. Derived classes like DNA sequence and protein sequence inherit those rules automatically and then add their own specific biological logic. This hierarchical structure means bioinformaticians don't have to rewrite millions of lines of foundational code every time they analyze a new type of molecule. Biology also uses conserved interfaces. Different proteins might share the exact same structural motif, but they execute entirely different chemical functions depending on their cellular environment. C++ handles this behavioral flexibility through polymorphism. You send a single command to two different entities, and each reacts according to its specific chemical nature. If you call a method like calculate mass on an array of mixed molecules, the DNA object calculates its mass using nucleotides, while the protein object calculates its mass based on amino acids. The single command adapts to the object receiving it. Inheritance and polymorphism give bioinformaticians the power to evolve their software architectures with the exact same efficiency and adaptability that nature uses to evolve organisms. When you combine these four pillars, classes, objects, encapsulation, and polymorphism, you transition from theoretical computer science to applied biological engineering. Modern biology relies on object-oriented databases. Instead of flat text files, these systems treat sequences and mutations as an interconnected ecosystem. This architecture allows toolkits to search terabytes of data and isolate a single cancer-causing mutation without crashing. The completion of the Human Genome Project mapped our DNA and permanently shifted biology out of the wet lab and into the realm of computational science. Managing datasets that double every few months requires the same level of architectural foresight used in large-scale infrastructure. By structuring code around DNA's instructions, the protein's physical presence, and the cell's protective boundaries, we keep our computational models coherent at any scale. Mastering bioinformatics requires designing modular architectures that can survive the unpredictable growth of genomic data. When our software adopts the modular, encapsulated logic found in the genome itself, it stops being a simple processing tool and begins to reflect the actual, breathing architecture of life itself.